Good morning, everyone, and a warm welcome to the International Conference on Frontiers in Industrial Biotechnology, hosted by the Department of Biotechnology from St. Joseph's College of Engineering. Before we start, I request you to follow a few instructions to ensure the smooth sailing of the event. Please do not unmute yourselves at any point during the session and do not introduce yourselves in the chat box. Kindly post any questions you have in the chat box and they will be answered during the Q&A session in the end. If you face any technical difficulties at any point, relay them in the chat box and we will look into it. We will now begin on an auspicious note with a Tamil Thai Valthu. I now call upon the coordinator of the event, Dr. G. Kalavati, will now deliver the welcome address.
Good morning, everyone. On behalf of the organizing committee, Department of Biotechnology, St. Joseph's College of Engineering, I welcome you all to the AICT-sponsored International E-Conference on Frontiers in Industrial Biotechnology. It is indeed a great pleasure for me to welcome the speakers of today's session and all participants, including faculties, entrepreneurs, research scholars, and students from various places across the country and also from various countries. I also extend my warm welcome to the head of the department, my dear colleagues and students to the International E-Conference on Frontiers in Industrial Biotechnology. I'm sure that this International E-Conference will give you a strong insight into the recent research and cutting-edge technologies of the frontiers in industrial biotechnology through the plenary lectures from the eminent scientists and professors. Also, this conference will act as a platform for the young minds to share their research works through oral presentations. Once again, I'm glad to welcome the speaker of today's first session, Dr. Anik Luke Dhanaraj, Senior Competitive Intelligence Analyst, Bayer Crop Science and University of Agricultural Sciences, Bengaluru, India. Mr. Manjunath Rao, Head of Quality Management, Bayer Crop Science, Hyderabad. You're welcome, sir. Now I request our Head of the Department, Dr. G. Srikumar, sir, to give the inaugural address.
Good morning, everybody. I'm extremely delighted to proclaim that the Department of Biotechnology is hosting a 3-day international e-conference on frontiers in industrial biotechnology between 27th and 29th July 2020, which is sponsored by All India Council of Technical Education, that is AICTE New Delhi, India. In this moment, I wish to brief some highlights about our college and the department. St. Joseph College of Engineering, one of the top institutions in India, with the 160th position as per the current National Institutional Ranking Framework. It has got high reputation in educational society for more than 26 years, and now it has got NAC A+ grade accreditation too. It is having 10 UG programs and 7 PG programs with an affiliation from Anna University, Chennai. Our Department of Biotechnology is 1.8 decades old, having the BTech and MTech biotechnology programs in it. So far, we have bagged 139 university ranks with 10 gold medals and 7 silver medals with 100% pass percentage every year. The department has got state-of-art, high-end, sophisticated laboratories having more than 1.1 crores of rupees as government funding for conducting research. We have also fetched a research center recognition from Anna University to do PhD and MS by research programs. Current COVID outbreak and subsequent lockdown make us inert by both physically and mentally. This problem is more realized, especially by the students and academicians community, because all the time we were mentally more active when compared to others. In my view, high priority should be given on making our biotech community abreast with the current ongoing research trends and not allow them to lose their interest in the subject. The uniqueness of biotechnology is its multitudes of areas of research. There is an incredible skill, knowledge, and research advancements are available among those diverse research communities. A conference brings them in a common platform and connects each other. It would provide an affable environment for the academicians and industrial experts to freely exchange their views with actual practicing technologists. When you improve your knowledge, the knowledge itself will improve you further. This 3 days international conference is a best chance to achieve it. We have gathered a global level biotech family of people who will replenish our scientific resources and bring new visions to shape up our future research too. For this, we have a spectrum of intellectuals who are going to address for these 3 days international conference sessions. Dr. Anik Luke Dhanraj, Senior Competitive Intelligent Analyst, Bayer Crop Science, University of Agriculture Sciences, Bengaluru. Dr. Zainul Akmar Zakaria, Associate Professor, Department of Bioprocess Engineering, University Technology, Malaysia. Dr. Jose Antonio Teixeira, Professor in Industrial Biotechnology, University of Minho, Portugal. Dr. Sanjeet Meharia, Department of Engineering, University of Campania, Italy. Dr. Prashan Kumar, Postdoctoral Fellow, Department of Chemical Engineering, Chunbuk National University, Korea. Dr. Sankeet J. Joshi, Deputy Director, Oil and Gas Research Center, Sultan Qaboos University, Oman. Dr. M. Prasad Vittal Rao, Scientist, Bioenergy and Energy Planning Research Group, Switzerland. These 7 plenary lectures are going to cover a potential industrial biotech research areas such as innovative products for farmers and seed quality testing, biotechnological applications of microbes, clean energy for production of chemicals, nutraceuticals from microalgae, polyhydroxyalkonates as natural polymers, role of biotechnology in petroleum industry, biofuel sustainability assessment, etc. In addition, we have technical presentations by the participants also. In order to moderate and assess the quality of the presentation, we have a combination of external member and internal experts from hosting department as panel of judges. Dr. Anik Luke Dhanraj and Dr. G. Srikumar, Dr. Zainul Akmar Zakaria and Dr. G. Bhaskar, Dr. Sanjeet Meharia and Dr. Justin Pakia Jacob, Dr. Prashan Kumar and Dr. L.F.A. Anandaraj, Dr. R. Ravi Kumar and Dr. M. Chamundeshwari, Dr. M. Prasad Vittal Rao and Dr. V. Renuka, etc. At this moment, I appreciate the overwhelming interest shown by the participants. There are 363 registered participants. That includes 102 faculty members, 207 UG and PG students, 45 research scholars, and 9 industrial persons. These participants are from 4 different countries such as Kuwait, Saudi Arabia, Ethiopia, Oman, etc. and 16 states across India, from Jammu Kashmir to Tamil Nadu. There are 138 abstracts received for presentation. Out of that, only 90 abstracts are selected for online technical presentation, and balance 48 abstracts are taken for recorded offline presentation and posters. With this proud note, on behalf of the patrons of the event, Dr. B. Bob Manoharan, Chairman, Ms. Jessipriya, Managing Director, Mr. Sashi Shekhar, Director, Dr. V. Seshagiri Rao, Principal, and Dr. Parvatavardhini, Dean, I inaugurate this AICTE sponsored e-conference on frontiers in industrial biotechnology. As a mark of inauguration of this international conference, I take pleasure in releasing this conference proceedings. Hope all of you are seeing this. This will contain complete details about the conference, felicitation messages from organizers, technical lecture contents from invited speakers, research articles of presenting participants, and other valuable informations. I make a note that this conference proceedings will be sent to you for every registered participants along with your participation certificate at the end of the conference. I wish to convey my gratitude to AICTE New Delhi for sponsoring this international conference. I also extend my warm felicitation to the organizing members, Dr. G. Bhaskar, Professor, Ms. G. Kalavati, Assistant Professor, and Mr. R. Naveen Kumar, Research Scholar, and my dear final year biotech student coordinators. I'm sure this international conference will definitely provide an excellent opportunity to all the delegates to get more information on the latest biotech developments. Our present generation carries more responsibility than any other generation that lived before. And you, the participants of the conference, are at the forefront. When the entire world has slowed, we will rediscover ourselves. Let us learn together and even when we are apart. All the best. Thank you. Over to Ms. Vatika.
Thank you for your kind words, sir, and for releasing the proceedings. We will now display a closer view of the conference proceedings. This will be sent as an e-copy to the participants along with your certificates via email. Now, without any further ado, let's get into today's session. The first session will contain two speakers. It gives me great pleasure to introduce the first guest speaker for this lecture, Dr. Anik Luke Dhanaraj. Dr. Anik Dhanaraj is a science fellow from Bayer Crop Sciences, currently working as a competitive intelligence analyst. He has been working with Bayer for over 16 years and was involved in supporting the launch of biotech traits in India. He led the teams involved in molecular testing of GMOs needed for regulatory approvals in the country. Prior to joining the Monsanto, which is now Bayer, he was a visiting scientist in the United States Department of Agriculture in USA, where he worked on the genomics of cold hardiness of blueberries using microarrays and EST markers. For his PhD, he worked on molecular markers in cashew, tissue culture of jasmine for his master's thesis, and has over 25 scientific publications in international journals to his credit. Our next speaker is Mr. Manjunatha Bhimarao. He is the head of quality management in India. He has over 25 years of expertise in seed quality and product supply. He has been heading the quality management activities at Bayer for over 9 years, and is based out of Hyderabad. Prior to this, he was the quality assurance manager for South Asia in Syngenta Seeds. He has also worked in Indo-American hybrid breeds for over 4 years and has held different leadership roles during his career. He has a master's degree in agricultural from University of Agricultural Sciences at Gandhi Krishi Vignya Kendra in Bangalore. Once again, it is an immense pleasure to have you both with us today. I request you to take over the session.
Thank you very much for the very warm introduction. And nice to hear from Dr. Sri Kumar, and also thanks Mr. Bhaskar and Dr. Bhaskar and Ms. Kalavati for the invitation. So I think I'll just go to sharing my screen now. Okay. So good morning, everyone. It's indeed an honor and a privilege to come here and share with you all some information that we have gathered over the years in biotechnology and to see how it's applicable in various areas. Okay, so this session, which is about an hour, will be handled by myself and my colleague Manjunath, who's based out of Hyderabad. I'm in Bangalore. So I'll take the first 30 minutes and he'll take another 25, 30 minutes after that, totally about an hour. We'll leave some time for Q&A, question and answers, okay? So I'll be talking to you all or giving you an overview of the different technologies that we use here at Bayer to develop crops or to improve crops. And the main key message I want to drive down is, especially to your biotechnology students, it's to help you all realize that biotechnology is a wonderful tool to help to improve crops, but it's not the only tool. And it's very, very important that we learn how it is used along with other technologies out there to make products for farmers. And that is like an overview that I'll be talking about, to show you all how in Bayer we use not only biotechnology, but biotechnology supplemented or complemented with other technologies to make products for farmers. Okay, so this will be the overview of my presentation. I'll first give you a brief overview about what Bayer is, okay, and then why we are in agriculture, and the different platforms that we use to make products for farmers. That is breeding, biotechnology, our chemistry is biological, and then a little information about Bayer scholarships and things that usually interest students, okay, because most of you are students out here. And after me, Mr. Manjunath will take over and kind of talk about how the seeds or the ultimate products that we have are tested before it's released out to the public. Okay. Okay. So this slide gives you an overview of the entire organization that is Bayer. So Bayer actually has three main businesses. One is our crop science business. Okay. Now, crop science is the part that I'm showing you over here, where I'll be focusing on. So I'm working on in crop sciences, where we are actually use various chemical, biological, or various seeds and traits or digital technologies to make things for products for farmers. We also have a very strong pharmaceutical business. Okay, the pharmaceuticals is where we make prescription products, again, mostly around cardiology, cancer, oncology, and women health products. We also have a very strong consumer health division, where we actually make medicines that can be bought over the counter. Okay, so today's presentation, I'll be focusing mostly around crop sciences and showing you how we here at Bayer use biotechnology and other technologies in this area. Okay, so moving on, just to give you a background of the organization. So Bayer last year, our total sales were about 43.5 billion euros. Okay, and about 5.3 billion euros of this was pumped back into research. And why I'm highlighting this is just to get you all aware that how important we consider R&D and how much money we invest in R&D, because we know that it's only when money is invested in R&D can we be developing new products out there for our consumers. So almost about 1/10 of our sales worth goes back into R&D. We have about 100,000 employees, but 1 lakh employees globally, and we operate out of 87 different countries. Okay. Now coming to agriculture, I'm sure all of you will agree with me that we are all facing a challenge now. Okay. Soon by 2050, we're going to be about 10 billion people in this world. I don't think I'll be around, but I'm sure most of you all will be. And this is a problem that we all have to face. And how are we going to challenge that? Okay. We're going to have 10 billion people, a lot more mouths to feed. And by that time, about 70% of our food that we eat will be coming from different sources and not necessarily only plants. More of us are beginning to eat more and more food from animal sources, that more cheese, more milk, more eggs, more meat. So not only are we going to have more people, but we'd also need to have more animals to feed these people. And what's happening on the other front? So we're going to have a great demand for increase in all the on food and food products. But what's happening at the other end? Okay, here we see that actually a lot of our land is actually becoming less. We have less and less arable land. Okay, and the ratio of arable land to people on this earth is actually decreasing over the years. It's drastically coming down. Okay, and we're going to have large amount of losses in agricultural produce. It could be in the field, it could be doing transportation and all that. So here you see a big demand of we need more food to feed this growing population that we have. But we're going to have a scarcity of land, we're going to have a scarcity of resources, we're going to have all those scarcities. So how are we going to deal with that situation? So here in Bayer, what we do in crop sciences is we try and see how we can shape agriculture to ultimately benefit our farmers and our consumers when without damaging anything on our planet, okay? By making sure that all the resources are used very judiciously and taking care of our environment. Here we use our different technologies like our seed and trait business technologies, our digital agriculture business and crop protection, how we use them either singly or in combination to tailor make solutions for farmers globally. So what do I mean by tailored solutions? Now, the requirements of a farmer in Tamil Nadu or in Chennai or in those parts would be very different from a farmer who's growing, say, in Punjab, because the environment is different. So that way, based on our global farmers all over the world, we tailor make our products for them or tailored solutions using these different technologies. Okay. So these are our main businesses in crop sciences. We have a chemical and biological platform where we sell herbicides, fungicides, insecticides. Okay. We have a very strong seed and traits business, and this is where all our biotech traits fall into place. Okay. We have a lot of biotech corn, soy, canola, oilseed. Okay. A lot of vegetable businesses. None of our vegetables are GMOs, are genetically modified, but our vegetable businesses also come under this and traits. We also have a digital platform where I'll talk a little bit about how we use digital in agriculture and also environmental sciences. Okay. So now I'll go straight into the subject. So I'll talk to you all about how we here in crop sciences use these different platforms to make products for farmers. Okay, so these are the 5 different platforms that we work out of. One is plant breeding, biotechnology, our chemistry, biologicals, and data science. And as I mentioned at the beginning, it's not only any one of these platforms, but how we use all these platforms with each other to make better products for farmers. And that is a message I want you all to understand better. Just being a pro in biotechnologies may not take you all very far, but if you all are able to complement other technologies with biotech, it's going to take you all a long way. Okay, so I'll start off with breeding and biotech. I'll spend most of my presentation on this and just a brief overview on the others. Okay, so introducing breeding to you all. I'm sure all of you all know what breeding is. Okay, now I like this analogy of Google Maps when I talk about breeding, okay, or traditional breeding. Now, Google Maps, I'm sure all of you all use Google Maps to give us directions how to go to A to B. The same way, when we want to make a plant or get benefit characters of a plant, we can use traditional breeding. Okay, and this could be something like any route that we can take to get from A to B when we're bringing in traits from the same species. Okay, but you see in Google Maps, it also gives you alternate routes, right? So there are alternate ways in which we can make about bringing hybrids in the market. One is using molecular breeding, which could be better. It again could be compared as another alternate route that Google Maps still tells you to take, go from point A to point B. It may be definitely shorter than the earlier route, but it's a different way. So molecular breeding that way is beneficial over traditional breeding because it can speed up breeding programs. And then there could be another way of improving, coming from point A to point B, by using genetic modification, where you introduce a gene. So you make a product a lot quicker, which will be another beneficial way. And the latest that we talk about gene editing, where you can bring products to the market a lot quicker. Okay, so these are just different ways in which we can bring about genes that we want from different organisms or different individuals of the same species into one individual to make something that we want. Okay, so this slide talks about plant breeding. I'm sure all of you all are very aware of this or are familiar with this. So had we not interfered with nature at all, our watermelons wouldn't have looked like what it looks today. About 300 years ago, this is how they were. But because of what man, and this is how corn was thousands or hundreds of years ago, Tinosynthe and mustard plant. But what we've been doing is either intentionally or unintentionally, we've been making or choosing plants that are beneficial for us or crossing them without knowing what we are doing. And this is what it resulted in today. So that is what traditional breeding also refers to. So here we at Bayer use traditional breeding to make resilient crops, to improve quality of any produce and also increase nutritional values. And all this is by just crossing plants who we know where they have desirable characters to make an individual that we want. Okay, these are just some snapshots of products that we have here in India. Vegetable business, all our vegetables are sold under the brand name of Seminis. They're all hybrids. We also have a very strong corn business here in India sold under the name of Decalb. And these are only products in India. Okay. We also use molecular tools, as you're aware, molecular tools to help us speed up our breeding program, where we can associate a marker with a trait and then see if that marker is there and then take only those plants forward. And this is what is known as genotyping. So before we go to the next stage itself, we can wait, take a little bit of plant material from the leaf, little leaf material, extract the DNA and see if it has the molecular markers that we want. And only if it has the markers that we want, we can take those plants forward. Thus, we are reducing so much of effort on resources and time. Okay, so this is definitely more advantages than traditional breeding because it's a lot quicker and saves time. We don't let the plants grow up to see their phenotype. Okay, then the scientists put their minds together to think if there's any other way we can speed up this process. Okay, is there any way we can look at a seed and see if a seed has the traits that we want, and only in that case sow the seed. Okay, that's when the company came up with what we known as a seed chipper. Okay, so the seed chipper is again made by scientists completely in-house, and how by collaborating with people from different departments, by the biotechnologists speaking to the engineers, they've come up with the seed chipper. So what basically the seed chipper does is, as you all can see over here, it chips off a portion of the seed. Okay, and like these are what you see on your left are soybean seeds. It chips off a portion of the endosperm, and that little powder that you get out of it can be used is enough to extract your DNA and then to see if it has the molecular markers that you want. And only if it has the markers that we want, we can actually go and sow those seeds and then get a plant that we want. And that's speeding up our breeding programs a lot better. And to the right, what you can see is how the chipper has been used on different crops. Okay, like on corn, soy, cotton. It's extensively used in corn and soy. We don't have a chipper in India, but we have a few in the US. And these are what are used to help us speed up our breeding programs. So basically, what a chipper does is it chips a seed, and then we don't go and looking into the molecular marker analysis of all these seeds, the DNA fingerprints. We don't go and sow all the seeds. We just go and choose a seed that we want, that has the traits that we want, and that saving so much of effort and resources and speeding up our breeding programs. It is by using this method we can speed up our breeding programs a lot quicker. Okay, and with the evolution of PCR, it just helps us only to make our high testing platform more throughput. Okay. Now, when I was doing my PhD, my master's 15, 20 years ago, we used to PCR in tubes. Then we moved on to strips, but now we do PCR in 96 well plates. I'm not sure if you'll have seen these plates, but then we also do PCRs in 384 well plates, where you can put four times the number of plates, four plates, four times the amount of samples in this one plate. And the advantage of this is we are having reduced volumes and speeding up, increasing the throughput. And now here we at Bayer, what we do is we do PCRs in tape. Okay. And this doesn't go into a machine, but we go back to the old method of doing a PCR in a water bath using this tape, using this equipment down over here. And all this is just to tell you how we can harness these different technologies to speed up our breeding programs. Okay. And because of this, and I'll now at this time I'd like to just show you all the video on the seed chipper. So yes, so I'd like you all to have a look at this video, and all these videos are on YouTube, so you all can go. So decaf is the variety of corn a hybrid of corn that we make.
13 million. That's how many corn seeds we chip every year. An intense journey of genetic analysis and scrutiny with one unwavering objective: find only the best of the best. Those with the perfect fingerprint of DNA and the promise of maximum yields for the footprint of your acres. It's this proprietary process, among many others, that fuels continued innovation to bring new products to market well ahead of our competitors and greater selective latitude for local breeder testing. Ahead of the pace, ahead of tomorrow. It's one more look at the future of farming, already growing at DeKalb. Strong past, powerful future.
Okay. Thank you, Swastika. So I think I'll go back to presenting now. Okay, so that short video that that you all had a look at is showing you about the technology that we use that was developed in-house on the seed chipper. Okay, and what the seed chipper does is it helps us chip out a portion of seed without destroying the embryo, and then using that for analysis and taking that forward. So with this is how we use or enhance our breeding programs, where you can see I explained to you how we use certain plant biology tools or molecular tools or biotechnology tools to enhance our breeding programs, like using DNA fingerprinting and markers and all that. Already showing you all how we can integrate these different platforms or different technologies. Okay, so now moving on to biotech. Okay, so since all of you all are biotech graduates, I don't think I should be telling you all much about this because you all must be knowing everything, but just a very brief overview. Okay, biotech, GMOs or genetically modified plants, organisms have been in the market for more than 20 years now, like from the mid 90s. Okay, like in the US, these are some very popular GMO crops that are grown, like cotton, sugar beet, soy, corn, canola. Okay, and what you see over here are the percentage of acres that is being grown in the US. Okay, and the what you see over here, IR, these are the traits. IR stands for insect resistance. So these particular cotton plants are resistant to particular insects. HD stands for herbicide tolerance. So these are the traits that have been incorporated into the cotton genome, or here in the case of soy. Okay, I mean sugar beet. So this is just an overview of GMOs. All these are not Bayer products, but just an overview of GMOs in the US. Okay, and this is an oversight of how the different countries perceive GMOs. Okay, now when it is fully green, these are the countries that are open to growing and consuming or doing anything with GMOs. Okay, when you see the countries that are on orange, they are the countries that are open to importing GMO products, but they're not okay to grow GMO products. Okay, and the countries you see in this dark blue, they are the countries where you can conduct GMO trials, but under a lot of approvals. Okay, so it's still showing that a lot of awareness is required for the different parts of the world on GMOs. Now, the countries in orange realize the potential of the crops, but are only okay with consuming the products or importing the products, but not to grow them. Okay, so we need to do a lot to help them change their minds or help the others realize the benefits of this technology. Okay, so when we talk about GMOs, so in breeding, what we were talking about, how we can bring about genes from different individuals of the same species, but in biotechnology, it overcomes that barrier of species. Now, if I want to bring a gene from a microorganism that has a beneficial trait into a plant, this is how I can bring it using GMOs. So in India, Bayer has two products, that is had Bolgard, which is phased off now, and Bolgard 2. They are the only GMO products that the government allows us to commercialize or sell in India. Okay, and these products came in like in 2004 and 2006. Now, Bolgard was made by using agrobacterium transformation, by growing agrobacterium with the gene that was obtained from Bacillus thuringiensis to give insect resistance. Okay, and then you co-cultivate it with an X plant and then make sure it goes into all the cells, get a whole plant out of it. This is how you make a GMO. Whereas Bolgard 2, the X plant from a Bolgard was bombarded with Cry2AB2 gene, which gives a resistance to a wider range of lepidopterans, okay, and developed the Bolgard 2. And this technology, basically what it does is, these are the called bolls, or the fruits of cotton. And the white, what we see is the lint that we want. Okay, but this nasty worm called the boll worm comes and attacks the fruits right when it's about to fruit and destroying all the lint, making the corn waste. So more than about 40 to 70% of the cost of cultivation of farmers in India goes into control this pest. So what was done is they've identified a gene from Bacillus thuringiensis, put that into cotton. So when that gene is expressed in these cotton tissues, it'll produce the insecticidal protein that will kill this insect. Okay, and this shows the power of the technology. This is a photograph of a cotton field, a GMO cotton field to your right, Bolgard 2 cotton field, and the left it is a non-Bolgard 2 cotton field from an aeroplane. So here showing you all the cotton has been destroyed by the insect, where here it is not. Showing you all the power of the technology. Okay, and what it has done in India. Okay, the technology came into India in 2004. Okay, and right up to then, India was actually importing cotton from other parts of the world. But with this, cotton now being GMO cotton, now about 98% of the cotton grown in India are GMO cotton. Okay, and it's made India now one of the world's biggest producers of cotton. And overall, what's it done? It's changed the lives of millions of farmers. Okay, given them a lot more value to their life because they were able to get a lot more revenue. They were able to have better lifestyles, educate their families and stuff. Just showing you the impact of the technology here in India. Okay, now moving on. So all along, we are talking about how we need to be innovative in order to be able to produce more from less. Way back in the 1940s, about so much land was required to get about 10 bushels of corn. But in about the 1980s, about 40 years later, less amount of land was required to give the same amount of corn that we got from this yield. And all this was because of the Green Revolution and how people identified hybrids and started benefiting the benefit, started reaping the benefits of hybrids. And then came along GMOs, where you can introduce genes that you want, still getting more out of less, and having lesser land to produce the same amount. Okay, and now how we are seeing how we can use different technologies to see how we can make this land amount even lesser to produce more by using other technologies like gene editing. Okay, I'm not sure if you all are aware, but in Bayer, we are also using breeding, biotechnology, and also gene editing to make shorter corn. Now, what is short corn? Short corn is usually corn can grow up to like about 10 feet, but the disadvantage is when there are high winds, they break. But if you can make the same corn be short, but produce the same amount, it has various benefits. Okay, so that's our new product that we have that is going to be coming out soon, where we use both biotechnology, breeding, and gene editing to make these products. Now, again, I have a 1 minute video. So, Swastika, back to you for the last video that just talks about the how we use these different technologies in making short statured corn. So, Swastika.
Oh, yes, sir. Give me 2 seconds.
Yes, sir. Yes, so this is the last video that I'll show you all, then I'll come back to the presentation. So as I was telling you, this is a video that talks
Agriculture
about how
is the new front line. Farms are the new climate frontier. Innovation is the new sustainability. And short is the new big thing for corn. We're exploring breeding, biotech and gene editing approaches to do even more with less. Growing corn that's shorter, thicker and sturdier, stalks that can stand up to harsh winds and challenging weather conditions. Reducing crop loss, conserving resources, helping farmers protect crops and the planet. Field tests are happening now. This is just one way we're shaping the future of agriculture.
Yeah. Thanks, Swastika. Yeah. So what you all just saw is about how we use different technologies, like including gene editing, to make short statured corn. Okay, now let me go back to presenting. So can you all see my screens now?
Yes, sir.
Okay. So to just sum up what I've been talking about here is how we use different technologies to make products for farmers. The first being what I was talking about is breeding. Okay, let me just get my pointer. Yeah. So how we can use breeding to make crops better by introducing genes from different individuals of the same species. We make cross selections to make better hybrids. And then when we have identified traits through gene editing, I mean, not through through GMOs from other organisms, we can incorporate those biotech traits into superior hybrids and making them even better. So how we are complementing the technologies from two different platforms. Okay. Moving on to crop protection. We also have a crop protection platform where we sell herbicide, insecticides, fungicides. Okay, and these are again used used and made depending on the needs of farmers all over the world. Okay, and we also have biologicals and some chemicals that are used to coat seeds and that grown on the outside the plants. All this time I've been talking about how we use genetics and the other things that are within the seed to make better products for farmers. But here I'm talking about products that are outside the seed to when the plant grows, to keep them strong and healthy. Okay. And then these are the biologicals that we collaborate with other companies that have a strong in biologicals and see how we can leverage their technologies on increasing the value of our products to enhance the growth of our hybrid of our seeds and stuff like that way. Okay. And last but not the least, how data is shaping everything in agriculture. Okay. And you'll be surprised how data science can help us improve or make better products for farmers. Like in seed genetics, how we can do predictive breeding by using a lot of historical data instead of going and doing so many crosses in the field. We can use that to do predictive breeding. And Bayer bought over a company called Climate a few years ago, and that company has a lot of information on climate. So using all this historical data, what we can do predictive analysis. So these are in farmers in other parts of the world where their land holdings are large. Okay, so they have large land holdings. So we can, when they buy our products, we can also give them a prescription that can tell them most likely it's going to be raining 10 days from now. So the likely date for you to take up sowing would be this date or this date. And using sensors to actually evaluate the fertility of soil. because their land holdings are large, we can tell them you need not put a blanket amount of so much nitrogen everywhere, but in certain pockets where it is needed, all based on sensors and information that we get from satellites. Now, in India, it's a very, very different scene. We have a smallholder farmers, right? So what we do here in India is we have an app called FarmRise, that actually this app farmers can download them, and and it gives them information to farmers on different crops that are available and the cultivation aspects of different crops, and also tells them another very important thing is on the cost that where they can sell their produce in the markets nearby. Okay, so this is what we are doing here in India. Okay, and how do we do all of this? All this we do out of with lot of responsibilities. Okay, we all the work that we do we are very, very transparent. We are open to dialogue and education, so people want to come and listen to us to see what we are doing, they're welcome to come and see what we are doing. And this is how we work. Okay, so these are the efforts that we are taking over here to see how we can bring about products for our farmers in a very transparent and efficient manner. Okay, and this is a slide I like to flash to students because it is some benefits that the organization has for students out there. The organization has various scholarships that are available for students pursuing higher education. Okay, they also have a youth summit where young people like you all can come up with wild ideas on using biotechnology or gene editing or anything, and how that could be made into, and if it is selected, you can go and present it there and you'll be given some sort of award. And how you all can even partner with NGOs or the organization on any of these uh, ideas that you all have. Okay, so that's what I had. And if any one of you all are in Bangalore, please, we have a R&D in Bangalore. Please stop by, come and visit our labs. We call it a Bay Lab, or even if you all are open to, we can give you all a virtual tour of the lab from there. Okay, and this is our website where it tells you a little bit of what we do over there. You're most welcome to come there and join. So all this time I've been talking to you all about how we make seeds, okay, how we make products for farmers. So now I'm going to hand over to my colleague Manjunath is going to talk about how they'll test these seeds before they get them to the market, and after which we're open to Q&A. Okay, so with this, I'm done. So I'll hand over to Manjunath. So I'll take questions after this. Thank you.
Yeah, thank you, Dr. Anik.
Yes.
First of all, thanks for giving me this opportunity. Thanks to organizing committee and also respected faculty and my dear students. So today I will take you through what in Bayer seed quality testing we do to maximize the benefits to the our farming community or our key customers. Okay, so what we do in Bayer. So the main mission statement for the quality testing organization is to provide relevant test. It's not that a prescribed number of tests. We will going to do only the relevant test, and we do it in timely before the material reaches to our customers, and we try to provide the high quality product and services, and the tests are cost effective. Okay, it cannot be tested with a very high cost. Okay, because we are dealing with some of the crops with marginal ROIs also. It's all based on the what type of test is required and with what cost. And final aim is to provide the best quality seeds to our customers and also the best services. So having said that, our main motto is to do what is quality in all we do, whether it is the production, whether it is the processing, whether it is the packaging, or it is the quality testing, or the final, whatever the marketing and the commercial activities we do, it is with a motto of quality in all we do. Okay, so globally we have 4 regions. Okay, in the South America we have few laboratories. Mainly the key laboratories are located in North America. So where we have the innovation engineering team sitting over there. So they will be doing a lot of research on the seed production innovations, whether it is the production or the SPR or for the seed quality testing. Okay, and coming to Europe, we have so wherever the tomato photo is depicted, it is the vegetable laboratory. Wherever the corn is there, it is the row crop or the field crop laboratories. Similarly, we have in South America 3 laboratories, both for row crops as well as for the vegetables, and we have a lot of laboratories working in Europe as well as for the South Africa. And in coming to Asia Pacific, we have one lab in Beijing. In China, it is mainly for the vegetables. We have another laboratory in Lahore, which is mainly for corn. And similarly in Pitsanalok, which is in Thailand, it's mainly for vegetables as well as for the row crops. In India, we have both for vegetables as well as the row crops. So in India, we deal with two row crops, that is corn as well as rice. Whereas in Pulilan also we have the row crop seed quality testing facility, which is mainly for corn as well as for rice. Okay, today what I will going to give a high level overview of what is meant by seed quality testing techniques, and which is mainly the comparing the age old techniques as well as what are the new methods of seed testing in physiology. Physiology mainly includes the germination, vigor test, physical purity and physical appearance. Mainly these are the physiological tests what we do. And then the genetic purity, and we will discuss about what is genetic purity, and the seed health mainly in the vegetables. In physiology tests, mainly it is the physical appearance and the physical purity part. So mainly in the old method, a old testing method is mainly to use the purity boards to analyze the physical purity and physiological analysis. Now videometer is the latest technology what we are using. And then the seed moisture test, which is used to be a woven method, which was time consuming and not reliable in some of the cases, and also it is a laborious process. Now we have come up with the latest seed moisture meters, which gives the seed temperature also apart from the seed moisture. Whereas the germination test is usually comprises of the roll towel method or the paper method, we used to call it as, and also top of the paper, and also some of the vigor test like nursery emergence test, usually for the wet crops like rice, and also soil cold test. And we also do field emergence test in some of the dry crops. So these are all the very traditional way of doing the vigor test. And also in vegetables, we use usable transplants, which is a field emergence test. But what is there in the how to improvise on this test? Because it is, one is the turnaround time, so the older method was taking, and the second is the accuracy and the reliability and the repeatability. So now in Bayer, we are working on the videometer technology for all these three platforms, mainly for the seed physiology and also for the seed health, where it will work on the high precision multispectral imaging. Okay, so it detects the physical qualities of the seed, right? What is the size of the embryo? Where are the internal cuts on the seed? Okay, so, and also what is the vigor? How, because we correlate the physical appearance and the size of the embryo with the germination as well as vigor. And also using the staining technologies, we also working on seed health test. What are all the pathogens which is there in the seeds? Okay, so this is not only helps in detection, it also helps in the automation of the technology, where we don't need to enter the results either on a paper and feed it into the system, but it directly detects, and through the algorithms it directly enters the results will be entered into system. Okay, and also these, most of these methods are non-destructive, and also it is very easy to handle in the laboratory. So I would like to draw your attention on the vigor. So germination is most probably everyone knows. So germination is very straightforward. How much out of the 100 seeds is germinated under a favorable set of climatic conditions. So usually we do it at 25 degrees centigrade plus or minus for most of the vegetable and row crops. Whereas vigor is the one which takes lot of attention, because most of the seed quality complaints come in terms of emergence, because India being mostly a rain-fed agriculture, and around 70% of our row crops especially grows under rain-fed conditions. So it is subjected to lot of climatic adversities, temperature adversities, or the moisture adversities, where the vigor of the seeds comes to is very, very important. So the old method was usually to seeds are planted in a wet substrate, either it is in soil, sand, or in other media, and placed in cold environment to test the vigor of the seeds. Okay, and then it used to be transferred to the favorable condition and manually evaluate the normal seedlings, abnormal seedlings, dead seeds, like that. Okay, so it was all the visual observations we used to do to evaluate the vigor. So there are many vigor methods, but what I am talking about is the emergence test using the soil core test method, where it is a more of a manual observation, and all the results has to be entered manually into the system, which was a problem for the mainly for the transparency and also traceability. Whereas in the imaging technology, we call it as radical emergence test. We place the seeds on the gel, okay, and subject it to the cold condition at certain temperatures and for a particular set of duration. Or it depends on the crop. In corn it is different, in rice it is different, in few of the vegetables it is different. Okay, so it is grown on a media and analyze it through imaging technology. So it is fed into a imaging equipment where it measures the radical length using the high-tech photographs, and using the algorithms, it will convert that into the percentages. So it directly gives the vigor ratings in terms of percentage. So once it detects the radical length which is emerged in the gel media. So this is very much repeatable, and it directly correlates to the field conditions. Okay, so we stimulate those stringencies or adversities in the laboratory and do the vigor analysis. So another important test after physiology is the genetic purity. So usually the age-old technique is the phenotyping, where we used to compare the morphological characters through visual observations, because we have to grow the crop and assess individual plants on a particular time. Suppose if the physiological inspection is for the flower, we have to wait till the flowering comes. If it is on the hairiness, we will see the hairiness. If it is on the bold shape or the petal color, pollen color, so there are many visual observations we make in the phenotypic genetic purity, which is called as grow out test, which is time consuming. Depending on the crop, it will take 140 days. In a crop like rice, which is a long duration crop, it will take 140 days. Okay, whereas in vegetables, it may take up to 100 days. In corn, it may take around 80 days. But sometimes it will not help the business. Okay. And the second one is the protein-based markers, which is usually called as the isozymes technology, where we identify the isozymes, the isozyme markers, which is mainly based on the protein banding, and assess the genetic purity. Again, here the challenge is for some of the new genetics which is coming, it will be very difficult to identify the markers, protein bandings, and also it is not repeatable. So again, it is an outdated version. And then, so PCR techniques, PCR and RT-PCR, real-time PCR are the new generation DNA-based technologies, which Dr. Anik has explained, and also you are the biotechnology students. I will not take much time in explaining what is PCR technique. But one thing I would like to explain is the again advancement in terms of Tachman-based technology, where it is a high throughput inputs. Okay, we can assess the genetic purity very quickly for large number of samples. Okay, again, the one drawback here is we have to identify the markers to assess the genetic purity, whereas the future technology what we are working on is genotyping based on the sequencing, GBS. Okay, here the variety or the batch, we completely sequence the entire history of that particular parent or the hybrid, and we compare that with the commercial batches, so which is a much advanced method. Okay, but still it is not implemented. It will take some more time for the commercial implementation. Similarly, the seed health techniques has also changed a lot. So earlier we used to work on the blotter methods, where we used to grow the pathogens on the media and assess through the microscope to detect the pathogens. And again, the bioassay or the pathogenicity test, where we used to inoculate the plants to make advancement in the breeding programs. Okay. And similarly, ELISA is another test we usually used to do it for the viral detection. So which is an antigen and antibody related. So we get the kits for the ELISA test. So using those kits, we detect the pathogens. Whereas the advanced method, again, as I explained it earlier, so one is the PCR based techniques, where high throughput equipments we are using in Bayer to get the results very fast and with more accuracy. Similarly, we will be using the videometer technology for real-time data collection and also for the accuracy. Okay, so these are some of the techniques what we will be adopting in Bayer laboratories across the globe. One is to provide the best quality seeds and in time and with appropriate cost. So with this, I am closing my presentation. If there are any questions, Anik and myself will be ready to provide the answers. Okay, if not, we will get back to you with appropriate answers. Thank you so much for this opportunity.
Thank you very much, sirs. We actually have just one question from YouTube, and that is, when do you think these equipments will come to India?
Oh, so can I go with that to answer that question?
Yeah, Nick. Yeah, please.
Okay. So actually, except the chipper, all the equipments are already in India. Many of the companies, many even institutes use all the equipments that we spoke about, or at least most of them. So they're already here. I hope I answered that question.
Yeah, especially for seed testing, Anik, just I'll give you some background.
Yes.
So
yes, please.
This radical emergence test is very unique to Bayer. Okay, so this is a bigger test we have deployed in most of the countries for row crops and also some of the vegetables. Okay, and similarly for videometer technology, it is in the advanced stage of innovation and validation. So probably in a 1 to 2 year of time, Bayer labs will be using these technologies.
Thank you very much, sir. I'm pretty sure that answered the question that the participant had. Thank you very much I'd like to thank both of you for an informative session on developing products for farmers, as well as on the scholarship at Bayer. It will definitely be useful to many of us, especially during these trying times. Now I call upon the coordinator, Dr. G. Bhaskar, to deliver the thank you note.
Good morning, Dr. Anik and Dr. Manjunatha Rao. Good morning to everyone. So, yes, so it's a great pleasure for me to thank our today's speakers for session 1. Dr. Anik Luke Dandraj and Dr. Manjunatha. On behalf of the organizing committee, International Conference on Frontiers in Industrial Biotechnology, I wholeheartedly thank our speaker, Dr. Anik, Senior Competitive Intelligence Analyst, Bayer Crop Science Bangalore, for accepting our invite to give a plenary lecture and enlightening us with the advanced technologies used in Bayer for crop production. Also, I am thankful to Mr. Manchinatha Bimara, Head of Quality Management, Bayer Crop Science Hyderabad, for joining with us in today's session and sharing wonderful information about innovations in testing on seed quality. So I thank both the speakers for spending their valuable time with us. So whenever you approach Dr. Anik during last 2 3 weeks, he has cooperated with us well for our schedule, and he has spent a lot of time for giving this plenary lecture. So, and I thank all the participants for your kind cooperation throughout this session. Once again, I thank our speaker, Dr. Anik, and Dr. Manjunatha. So thank you, sir. So thank you for joining with us and gracing this event. Thank you, sir.
Thank you, sir, for having us over. Thank you.
And all the best for the rest of the 3 days. Thank you so much.
Yes.
Thank you, sir.
Thank you very much, sir. We will now move on to the second lecture of the day. The speaker for this session is Dr. Zainul Akmal Zakaria, who is an associate professor under the School of Chemical and Energy Engineering at University Technology, Malaysia. He has a Scopus H index of 17 and has published 2 research books, 9 edited books, and 13 book chapters. One of his research books was awarded the National Book Award in 2018 under the biochemistry category. At present, he has guided 4 PhD students as main supervisor. He has led various national research projects worth around 1.5 million ringgits and was involved as lead research in other projects with cumulative amounts of 3.7 million ringgits. He was a consultant for LJS Environment Australia, a guest editor for special issues in 3 ISI index journals, an evaluator for international research grant involving Australia, South Africa, Saudi Arabia, American Chemical Society, a visiting scientist to Argentina and Mexico, as well as was the program head for the UTM CONICET and an R&D research program at Argentina from 2015 to 2017. Dr. Zainul currently holds the position of associate chair at the school and is heading the UTM Bactec Research Group, where he works on biomass valorization and microbial technology. Sir, it gives us an immense pleasure to have you with us today. I request you to take over the session.
Thank you very much, Pratika. About that. So, hi everyone. My name is Zainul. I'm from UTM, University of Malaysia. So, I would like to thank my colleague Baska for inviting me for this afternoon session. And yeah, I would like to share a little bit about some of our experience, my research group experience working with bacteria mostly in various processes. So these are the list of my main research team. So just to share a little bit about the what UTM looks like. This is our main campus. This is where I am now. We have 3 campuses: Johor Bahru, Kuala Lumpur. Our campus is our satellite campus, and also one is small in area called Pago, which is focusing on agriculture activities. Right. So yeah, you're most welcome to come to UTM Johor Bahru anytime. This is my where I am now, the building. All right. This is where the School of Chemical Engineering is. So just to share a little bit about how do biotechnology is governed or is planned in Malaysia. So we have this body called Bioeconomy Corporation. So they are actually a body directly under the Ministry of Agriculture. So they are owned by the Ministry of Finance. They get the funding directly from Ministry of Finance, and they are actually responsible to plan. All right, to strategically plan and to structure the way forward for Malaysian biotechnology area. Yeah, and for the time being, these are the we are focusing on 3: industrial biotech, health biotech, and also agriculture biotechnology. So in line with the theme of this conference, so I'm going to share a little bit about Malaysia's industrial biotechnology. So these are the sectors or subsection available, which are the focus of Malaysian biotech, especially in the industrial. So hopefully this afternoon I can share a little bit about what we have done in two of the focus areas in biotech in Malaysia, which is bioremediation, which is focusing on treatment of organic industrial waste, as well as production of the application of microbiome technology to produce biofertilizer. Just a little bit of that. So yeah, this I think most of you are very well aware of the availability of these kind of techniques. Yeah, these are the more or less what we are trying to counter. All right, these are the least which is chemical based and also physical based. Also, this available here. So we are as a biotechnologist, we always try to find an alternative to this technology so that we can come up with a approach that's more or less should be more viable and also sustainable to what being offered here. Yeah, so here we are, we have the microbiome cell alternative. So either you are using different types of microorganisms. So in this case, I'm going to share a little bit about what we do with bacteria. So we have done some work in biosorption of heavy, we are mainly focusing on heavy metals and also some types of high strength of industrial effluent, for example, those that contain high organic content. And also, we also have some experience with bioleaching. Yeah, we try to get the precious metals out from the gold ores. Yeah, gold ores means refractory ores we're talking here. And also biological reduction. Yeah, in this case, this is what we have done for the past 7 to 8 years last time. Yeah, where we try to reduce the concentration, the presence of chromium 6 to chromium 3, and our targeted industry was electroplating, because the state where I am now is called the state of Johor. We have a lot of electroplating industry, and these are actually the small and medium scale industries, which are normally wouldn't have the capacity or the fund to have a complete water treatment system. So hence, we are working very closely with also the Department of Environment in this case. All right, just to share a little bit what we have done. Okay, biological reduction of chromium 6 using liquid fungible waste as nutrients. If I may share, we make it a point to always have this in mind, that to use also the nutrients, the agricultural waste as substituent for the nutrients in this process. Okay, so these are not the actual photo. I got this pulled by my students. The slide, yeah. These are the current techniques. All right, chemical based hazardous workplace, which they are using conventional water treatment plant, which normally would present a hazard of possibility of a chemical spillage. Yeah, hazardous workplace, definitely chromium 6 are carcinogenic and toxic, highly toxic. Okay. Even though at the end of the day it can produce clean final discharge, these are actually those that can afford it by the big industries. All right. When we're working with this project, we were quite lucky because we got the direct support from the biggest actually electroplating industry in Malaysia. So that is actually a good strategy, because you want to demonstrate what you have developed in your laboratory, maybe at the pilot scale. Is good, and is a good option if you can team up with the main player, because if you can achieve considerable success with them, it can easily convince other smaller companies. That's what we have done last time. So for the big industries, because they have big income, okay, they can afford sophisticated, expensive water treatment plant, so they don't have a problem with discharging their wastewater. It's just that there are also this cluster of industry. Okay, I'm sure in India also, in anywhere in the world, we have these SMEs. Yeah, they normally operate some even as a backyard industry. Okay, they don't normally have a proper WWTP, water treatment plant. So what do they do? They just dump. Okay, these are a fact that we need to digest and to try to come up with a solution for that, because they don't have an option. They don't have the money to buy the water treatment plant. proper water treatment plant. So these are the normal excuses, more or less like the answers given. All right. They don't have a choice. KA is actually stands for Quality Alam, is a company in Malaysia which is responsible to take care of these scheduled waste. All right. So they don't have a choice. They are even willing to pay the fine of 100,000 K. Okay. They are willing to pay that fine. Okay. So this is what we have done. We isolated a chromium resistant and reducing strain. Okay, because when we isolated it originally from this area, this is actually a dye manufacturing. We call it as batik. Batik is a type of cloth, a local cloth in Malaysia. It's a conventional cloth worn by in some events. Okay, these are the washing section. We got a sample here. Then we isolated a lot of resistant strain. Okay, surprisingly, but the one that can resist the toxicity of chromium 6 are not that many. So the one that can resist, we proceed with getting it into a pure culture. Then we preserve and work with it further. All right. So why do, as mentioned before, we work with liquid pineapple waste? Why? Because in Malaysia, we have a lot of pineapple fruits. Okay, we are not that big. We are only number 18 in the world in terms of production. However, the state where we are in, UTM is in, we have the largest. We are actually the largest producer of pineapple in Malaysia. So to get the fruits are not that difficult. Okay, these are actually the actual photo that we took from the plantation nearby. Okay, so what do we do with it? These are the solid pineapple waste. We dry and grind it, and we mix it together with our microbial cells, with our isolates, and we make it into the final form before we use it in our process. All right, these are just to show the lab scale first. What we did last time using just simple column, glass column. So we let it go up to 42 days, if I'm not mistaken. So we need around 2 weeks. Okay, 2 weeks to get 3 days. 3 days of continuous pumping of nutrients. Okay, we use here is the cellulose foam bunks. These are cellulosic materials, the commercial foam bunks. So we cut it, and we first of all we put the nutrient in, then we put our culture in nutrient broth in, then we remove we drain the column, then we pass through the liquid pineapple waste, because we gradually try to force more or less the bacteria now to get their nutrients from nutrient broth and switch to liquid pineapple waste. So we need a further 3 days for that. Okay, how do we measure? Is by the OD. All right. So this is just to show the development of biofilm, because along the line, when you start to pump the liquid pineapple waste, the biofilm will start to form. Okay, these are the photos, the SEM photos that we got. This day 40 shows the depthness. Okay, the biofilm start to thicken. Okay. Okay, for the liquid pineapple waste, this is where we got from the liquid pineapple processing factory nearby. So this is the raw pineapple, is actually acidic. The pH is around 2.8 to 3.2. Okay, it doesn't very seldom to go beyond that pH range. So it's not a good pH to grow microbes. So we have to bring it up the pH to 7. Yeah, but you need to do some pre-processing, pre-treatment of the raw pineapple waste, because it contains a lot of debris. Okay. Okay. This is actually the micrograph that we got from another column that we have. All right. This is the actual microbe that we have, the more or less that I can call the champion strain. After identification using 16 sRNA, it is identified as Acinetobacter hemolyticus. Okay, we deposit in a gene bank with the accession number of EF369508. Yeah. So what is unique about this strain is that it is an aerobic strain. Okay, Why is this unique? Because for chromium reduction, chromium 6 reduction, normally it is carried out in anaerobic condition. But in our case, we were more or less quite lucky because we got an aerobic strain, which gives unique features for our study. But since the normal one would be in anaerobic condition, when you use aerobic strain, it is at the expense of the biomass yield. All right. So this is the photo of our support system. Okay, in our case, one column we use cellulosic material. I mentioned just now we use the foam bunks. We also use wood husks. Okay, we try also gradually to move from using polymer to using natural materials, which also would have the capacity to be a good housing for the bacterial attachment, followed by the development of biofilm. So this is after day 1 week, and this is after 21 days. All right, you got a diverse microbial community over there. All right. However, after 61 days, we also make some analysis of the microbial population inside the biofilm here, which is shown here. So happy to say that our strain, the chromium-6 resistant and reducing strain, are still there. Okay, even though it has been, there are a lot of other strain presence, and by confirming that it is there, we can safely say that whatever result that we got is, we cannot exclude the contribution of other species. It's just that we can say that based on our work in the laboratory, we know that this strain can do the job. Okay, this particular asymptomatic Haemolyticus can do the chromium-6 reduction job, and it is contributing to whatever result that we got. Okay. Okay, what is this? Just to show that, the types of binding between chromium-6. Okay, chromium-6 in solution, chromium-6 present in the form of chromate. Okay, and also dichromate. It is negatively charged. So when binding, we always look for the positively charged functional groups on the bacterial cell surface. Okay. So we also carried out Zane's analysis, okay, XAFS, but we don't have this facility in UTM. We do this with our neighbor in Singapore, with National University of Singapore. So what is significant about this profile? Okay, it shows that during the whole reduction experiment, okay, we can safely say that because this arrow shows that recorded for dipotassium chromate, okay, other chromium compounds, okay, the binding, okay, it matches closer to chromium. I said we want to show it binds with what we have here, the chromium only, chromium metal, chromium nitrate, okay, the NO group, chromium acetate. Okay, chromium nitrate again. Then this is the chromium only, dipotassium chromate. Okay, it matches closer to the profile for chromium acetate, which supports our earlier FTIR finding. We show that it binds with the carboxylate group. Okay, in the bacterial cell surface, and also in this case, we can narrow it down that the binding means for the chromium-6 to be reduced, number one, it has to be absorbed by the bacterial cell surface. Then it will be translocated inside, because this is an energy-dependent process. Okay. After being absorbed, it will be translocated inside the bacterial cytoplasm region, where it will be reduced to chromium-3. Okay. What will happen after that is either it can be extruded back into the solution as chromium-3, or it will be deposited inside the bacterial cytoplasmic region. Okay, so in this case, from this analysis, we know that acetate plays a role in binding of chromate. Okay, chromium-6. Okay, also supports our earlier postulate, because if chromium-6 is binding with O from acetate, the The binding energy is like this. Okay, the interatomic distance is like this. But if it's bind with chrome 3, the binding energy is like this, which is what we got for our study. Okay, 1.97 angstrom. Okay, what does this tell us? Then after this, we also did some experiments, some evaluation at the 50-liter scale, of a bioreactor. 50-liter scale. We try to get the enzymatic fraction out from the cells. Okay, we produce some bits. Okay, so in our case, we were using an influent concentration of 60 milligrams per liter, which were initially mixed with liquid pineapple waste, that contain sucrose and also fructose. And some of you might ask, would it be that the sugar itself is taking care of for the reduction of the chrome 6 to chrome 3? Okay, the answer is yes and no. Yes, because it does to a certain extent, and it takes a long time. Okay, but with the contribution by the biofilm system containing our chrome 6 resistant reducing strain, the process is much faster. All right, and there is also the low possibility of system contamination compared to if you just leave it. You just mix the liquid pineapple waste with chrome 6. We also have a control experiment where even though you leave it there for up to 3 to 4 days, there are only a certain amount of chrome 6 that can be reduced by the liquid pineapple waste. Right? So the use of a biofilm system is really justified. In this case, after passing through the biofilm system, we pass the effluent through our chromate reductase beads. Okay, this is more or less should act as the tertiary treatment system. Okay, currently we have this ion exchange or activated carbon at any wastewater treatment plant, proper wastewater treatment plant. So we were investigating last time on the possibility of replacing the role of ion exchange and also activated carbon with chromium reductase beads. Okay, one thing about when you use reductase beads is that if you look here, the rate of reaction is nanomole per minute. All right, if you look here, the unit is milligrams per liter. Okay, that's why you need up to 24 hours of contact time. You need to recirculate the influent solution containing liquid pineapple waste, also chrome 6, into either your biofilm column or column containing chromium reductase beads. Because of this, right, the rate of reaction is much slower compared to the ability of the cells to convert 6 to 3 is much slower to what we are producing it, what we are giving it to reduce. In this case, is 10 to the power of 6 times. Okay, so what are the observation that we got? Why 0.05 milligrams per liter? Because this is the discharge limit set by the DOE, Department of Environment Malaysia, for standard B, for chrome 6. Okay, it's 50 ppb. Okay, and also we have determined that the enzyme is available mostly on the cell-free CFE extract fraction and not on the membrane fraction. And the process is enhanced presence of glucose. We have this liquid pineapple waste that contributes for the glucose concentration. And also it works best if it's carried out. The solution is at pH 7, plus minus 0.2 actually. And also these are the KM and Vmax. And also the process will be inhibited by the presence of silver and also mercury. Okay, what else do I have here? Okay, after we do this, we have proceed with the lab scale, 50-liter scale. Then we proceeded with a bigger scale. It's not that big. It's still a pilot scale. So now we already term our system as Chromeback. Okay, what is Chromeback is? It's a treatment system to remove chromium using bacterial technology that showed similar efficiency with conventional chemical-based approach at pilot scale. I'll tell you later why we put this statement here, similar efficiency. All right. So we have managed to test it at 200-liter pilot plant scale and also at 2,000-liter pilot plant scale. This is actually the actual photo showing that our plant on site, our industrial collaborator, which I mentioned earlier. All right. So some key features of our work with our 200 liter pilot plant is that this, remember the strain that I mentioned just now, Acinetobacter helminthicus. We use here totally wood husk, no more foam bounds, no more cellulosic polymeric materials. We just use wood husk that we got from the wood processing facilities nearby UTM as a supporting material. All right. When we grow Acinetobacter helminthicus, we only grow it in liquid pineapple waste. All right. So we sampled the electroplating wastewater from the industrial premise, and the concentration that we have is around 17 to 81 milligrams per liter from the wastewater treatment plant facilities. And the trend for, let's say, 1 month, there will be only 2 or 3 occasions where the concentration will spike up to 300 milligrams per liter. At this time, we don't go and take the sample because we know that this will be toxic to the cell. All right. So these are the flow rate that we use. Which have been optimized, and I'm happy to share that we have managed to reduce completely Chrome 6 to Chrome 3 immediately after the start of bioreactor operation. The start of bioreactor operation means after you have completed all the cycles of immobilizing the cells onto the wood husk. Then you follow it up with circulation of liquid pineapple waste. Remember the 3 days that I mentioned before? For this one, we prolong it to up to 7 days. Yeah, we just pump through the liquid pineapple waste just to make sure that the cells is attached and grow and hopefully form a biofilm. So after the end of that process, when we start to introduce anywhere between 17 to 81 milligrams per liter, why this is because this is the actual concentration we got from the electroplating facilities. We got a complete reduction directly. So this is actually a tremendous result for us. So after that, we follow it up with the conventional precipitation technique, because whatever we do here, we have to benchmark against the normal practice available at the industrial collaborators premise. So in their case, we are actually replacing the use of their sodium metabisulfite. Okay, they are using sodium metabisulfite to convert Chrome 6 to Chrome 3. So our challenge is to produce or to demonstrate that our humble biofilm system can also work like what they can produce at the factory. Okay, so these are actually the standard or the typical process which has been applied at the factory. Okay, and the last point here is actually very important. Even though we don't have any activities up to 10 days, when you activate your system again, it can take very short time for it to completely reduce Chrome 6 to Chrome 3. So this is actually a very important factor. Okay, so we also have got the system tested at 2,000 liter pilot scale. So what are the take-home points for both these systems? All right, so you can get a resolution of more than 39%, okay, provided the concentration is less than 300 ppm. Why? Because if you have 300 ppm, you can more or less take care of a big chunk of it from the presence of sugar from the liquid pineapple waste. Short residence times, because we use a downward flow in our bioreactor. We use a downward flow. We can get a complete COD, BOD, and odor and color removal. Definitely, this is actually complemented by the presence of the chemical treatment. And also, you only need chemicals for neutralization and precipitation. For this one, remember, we are benchmarking against the use of chemicals-based process. So this statement is actually a very big statement. Okay, and it's robust against fluctuation. The 70 to 81 ppm, you can tolerate the concentration. You don't need a very big space requirement because everything can be compacted. Okay, low nutrient requirements. Yes, we only need a maximum of 10 grams per liter. Okay, of glucose to be present. And also, since you are working together with DOE and their OSH unit, this system is okay, right? Of course, definitely you need to have more of the SOPs to make it fully compliant. And also because of we are using liquid pineapple waste, the sludge that we got, of course, it contains Chrome 3. it is also organic rich. Okay, we have tested it to be used as supplement for the growth of ornamental plant. Okay, if I can share here, these are those are the team last time. Most of them have finished their PhDs or masters. All right, we mix the sludge with the compost that UTM is producing. Then we are using it to as fertilizer to plant. In this case, we use balsam. I think they mentioned balsam anyway. Okay, I use balsam plant. Okay, so if you look here, this is the pot with the chromium sludge. These are the pot, the left one without the chromium sludge. Okay, so actually UTM is very happy with this. It's just that we are only at the pilot scale demonstration unit, so we cannot continuously produce and supply to UTM. All right. This slide is okay. We can skip that. Conservation potential. We can skip this as well. Okay. I don't know whether you're all interested in all this. Only, yeah. So, and now if I can mention, yes, no, or if I can mention at the end of the day, when we presented our case to our industry collaborator, our benchmark was how much sludge did you produce? Okay, they want to know that, number one, because when they are using their current treatment, which is chemical based, they are actually producing up to 15% sludge, no, 12% of sludge per month, which they need to send to the licensed solid schedule waste operator in Malaysia, and they have to pay a lot because they are very big. So in our case, we managed to deal with around how much here? More or less around 10,000 liters, because every time we take 1,000 liter, around 20 meter cubes. We managed to treat 20 meter cubes of electrolytic wastewater. We end up with 13% of the sludge. So they are producing 12%. We managed to produce sludge with complete safe disposal of electrolytic wastewater treated into the nearby drain. However, we produced 13% of chromium sludge. So they say that this is okay, but will not be improving their system that much. So they acknowledge that by using biotechnological approach, you can have more or less a similar system with which is also efficient. It's just that they give us an advice. Why don't you try to approach the smaller companies? Okay, that statement is very important for us to move forward. Okay, because an endorsement by the biggest electrolytic company in Malaysia to say that work with the smaller ones is the one that we used to discuss with DOE last time for enforcement. Right, it is still ongoing now. Okay, so that is about our. Okay. So the second example is we produce an improved version of biofertilizer that already there. Why? Because our biofertilizer now is specifically intended to improve the growth of oil palm and banana at the nursery stage. Okay, and for this, last time my PhD student was working with this. He was responsible to isolate the nitrogen fixing and phosphate stabilizing microbes from the oil palm plantation, and he is actually works at the oil palm plantation. He was doing a part-time PhD with me. All right. So yeah, these are not a very new data. Malaysia, I think, remain as the second world's biggest producer of oil palm in the world after Indonesia. Why so? Because if you look here, the trend of the oil palm plantation in Malaysia, in terms of million of hectareage which has been planted with oil palm, it is decreasing. Why? Because we cannot afford to allocate new lands for the cultivation of new oil palm trees. Okay, so it has been the target of the government. The target is two. Number one, with the decreasing, not decreasing, with the situation of less of new area of land for you to plant your oil palm. We want you to make do with the existing hectareage of land, but we want you to produce more oil, number one. Okay, means you have to do whatever to with the growth of the plant so they can produce more oil. Number two, tackle the issue of oil palm biomass. Okay, because for every palm oil that you produce, at the same time, we will be producing a lot of huge volumes, humongous volume of oil palm biomass. And in Malaysia, we have these types of oil palm biomass. The main one, okay, we have palm kernel shell, oil palm fiber, oil palm fronds, empty fruit bunch, and also palm trunks. And in this study, I'm going to share with you what we do with the empty fruit bunch. Okay, another one here is actually palm oil meal effluent. That is the single or the singular parts of the liquid fraction for the oil palm biomass. All right, these are the 6 main ones of the oil palm biomass that we generated. Okay, so what do we do? Currently, what do they do with the AFB? If you look here, they will mix it into the either mixture to produce paper, fertilizer, or also energy. Okay, when we were doing this work, we carried out this work. We were working with FELDA. Okay, FELDA is actually, where is my cursor? This is FELDA. FELDA is actually an acronym for the biggest oil palm planters in Malaysia. Okay, they are actually a government linked body who are being entrusted to take care of the plantation issues for oil palm in Malaysia. And all of these bodies, apart from FELDA, they are governed by this body called Malaysian Palm Oil Board. The acronym is MPOB. You'll see some word of MPOB later in my slide. So this is actually the actual photo that we got from one of the oil palm plantation that shows that now, especially for FELDA, they are going for a plant intercropping. They are choosing banana. All right, in between the oil palm plots, they are planting banana. Why? Because banana is also a highly a plant that requires a lot of inputs for fertilizer. So their normal practice now, some of the oil palm biomass, up to 50%, are being left at the plantation for it to degrade naturally. So rather than just leaving it there, some of it will be formulated into biofertilizer and be introduced to grow banana. However, in my study, we were focusing on how to improve the growth at the nursery stage. Okay. Okay, like I mentioned just now, oil palm and banana are among the highest nutrient demanding crops. So this gives us an idea of what? That if you can tackle the supply of fertilizer, you can help the industry a lot. Because up to 34% of the cost, okay, to process oil palm would be directed, or to grow oil palm would be directed for the fertilizer. Okay, so if we can do something to bring the cost down, people will be happy. Okay, so that's why we are targeting that if you can make sure that the plant grow faster at the nursery stage, okay, you can reduce the turnaround time for you to harvest the fruits, the oil palm fruits, and also the banana fruits, and you can more or less offset the cost of fertilizer because now you are improving the yield of the oil palm fruits and also the banana fruits. Okay, this is actually the actual photo in one of the oil palm mills in Malaysia. We have around 28 oil palm mills. 40 oil palm mills throughout Malaysia. The number is quite low considering that we are actually the second largest producer of palm oil in the world. So yeah, as I mentioned, we cannot allocate more lands, hence we cannot allocate more oil palm mills. So in this case, we are producing around 28 million tons of EFB per year, and also POME, palm oil meal effluent per year. Okay, that's a different story. Palm oil milk effluent. We also did study on this, but I'm not going to share it here because of the time constraint. So we're just going to share what we do with EFP, right? So we are all aware, the main component of a fertilizer is NPK, and also you need to also add components such as the major ones, all right? The macronutrients. Okay, this is not my student. This is his staff actually. So they were working actually together. Yeah, they also, if you look here, they also mix it together with POME, palm oil milk effluent, because it is still rich in organic content. For POME, the BOD can be as high as 80,000 ppm. Okay, these are actually, rather than you just consider it as a problem for you to treat, why don't you mix it, you mix it as a carbon source. Okay, you can mix it and use it for the production of biofertilizer. Okay, this is the actual photo that we got at the Tun Razak Research Station in the one state in Malaysia called Pahang. So these are the facilities that is available to us. So this shows that the banana seedlings, we do it like this. And if you look here, okay, 0% means normal fertilizer. 10% are the one that we are adding with our biofertilizer containing P3 means isolate number 3 that can solubilize phosphate, and 15 means, and 15 here means nitrogen fixing microbe, the 15 pure colony that we have. So what the choice of which microbe to work with, last time we narrowed it down to 2. Of course, they are nitrogen fixing and phosphate stabilizing capacity. And number 2, we base it on the fastest grower. Who will grow fastest? Okay, we have so many. So these are the strains that we actually choose. Okay, so if you can see here, after 16 weeks, there is really a marked difference between those that you add with the your newly formulated biofertilizer compared to the 0%. Those that you don't add biofertilizer. All right. The difference here is only the culture, the bacterial culture. That is only the only difference. The presence of POME, the presence of AFB, all are the same. But the difference is only the presence of cells that we isolated ourselves. Okay, this is for banana. The difference is not that big actually, yeah, compared to oil palm here. However, there is still some difference, yeah. So what was responsible for it? So this is actually the actual photograph, the SEM micrographs of our sample. Okay, A and B, C and D is after 16 weeks. I'm also quite surprised last time because the cell here looks more or less like the same species. Okay, I don't know what happened, but these are the actual photographs that we got. I'm expecting to see like a biofilm, you say, different diverse microbial species. So this could be one strain. Yeah. So yeah, we managed to show that our stuff that we put are still there. Okay. Just to show another micrographs of the, yeah, these are the name of the strain that we isolated. After 16 sRNA identification with Enterobacter closei, Burkholderia and Cepacia and Serratia marcescens. I know these are actually both are class 2 bacteria. However, these are also not a typical soil microbe. So we use it as it is. Okay. Okay, that is it for fertilizer. So the conclusion is that we have managed to propose to the Felda management about the potential of using this kind of approach. Yeah, this was actually completed 3 years ago. Okay, by my first PhD student. Okay, 3 years ago. So There are a lot of complications to bring it further forward, more or less because of the cost. Yeah, so the process is still there, waiting for it to be further supported. Okay, the third thing I'm going to share with you, I'm going to do this very quickly. I know time is not on my side. Is our biosorption work with gold last time, using AT ferrooxidants that we got from mining environment. Okay, very briefly, when we do FTIR study of the gold and AT ferrooxidant interaction, we focus on carboxyl and amine groups, and we can see that there's a more significant role of amine groups in gold binding, and this is fairly correct because gold in solution is in the form of negatively charged complex, is AuCl4-, and also the low concentration of amino groups in AT ferrooxidants. This is AT ferrooxidants. Concentration is around 0.98 compared to this one, glycine, due to the low amount of biomass that we are using. All right. Okay, and this is also supported by the binding between negatively charged gold complex, and also amine is also supported by this is FTIR results. We got that the amine stretching is much more significant, the difference compared to COO minus. Okay, which shows that in the presence of gold, which is a heavy element, the binding, the vibration is the vibration is shorter. Okay, because of the heavy presence of the gold atom. What else? Okay, we also carry out the voltammetric study. Okay, to check on the reducing potential of 80% towards AU3, whether ferrooxidants can reduce auric gold chloride ion. So we came to this conclusion that AU3 bind as AU3 plus. Okay, the significant role of amine groups is further supported by this study. Okay, and we also carry out some TM study. Okay, here you can look here. These are the control acetyl welfare accidents, and this is the micrograph showing the deposition of gold, AuCl4- as AU0 on the surface, mostly are on the bacterial cell wall. If you can see that, less presence in the cytoplasmic region. Okay, if you compare with the previous one for reduction, chrom-6 absorb at the surface, transport inside the cytoplasmic region, then either it will be extruded back into the solution or it will be deposited. Okay, but in gold case, in AU case, most of them will be deposited on the bacterial cell wall region. Okay, and those that be absorbed first would act as a nucleoside for more gold chloride to be deposited. Okay, this is also supported by X-ray study, which did not detect the presence of crystallized structure on the surface of bacteria, which shows that the gold is present in this AU0 from. Okay, what else? Oh, already conclusion slide. Okay, so to conclude, it can be said that microbiotechnologies are useful alternative for conventional physical-chemical treatment because you can still remember the whole idea of suggesting a biological approach is to provide, to give the industry an alternative. Okay, because you know that the commercially available are all either combination of physical, chemical, or chemical-based treatment. So we are targeting to produce also, if not standalone, to be incorporated into the existing treatment system, preferably at the tertiary stage. Okay, why tertiary stage? Because of the rate of reaction. You can still remember the nanomole compared to the milligrams per liter that they need to reduce, to absorb, or to convert. Okay. Second conclusion is that we have shown we can propose that you can also use agricultural waste. Okay, as raw material for various processes. In our case, we have shown that agricultural waste can be used as a useful substitute nutrients to grow cells, and also can form as the housing for the microbe in the biofertilizer case. Remember, EFB was used as the housing for and fixing also pea solubilizing microbes, and also for gold case, AU3 case. It doesn't. It's not for agriculture. Sorry. And however, you always need to conduct this to bring what you have in the lab at least to the prototyping scale, so that you can have a more significant trustability is more, because the scale is big but not big enough for it to be commercialized, right? And also you need to involve and get some support from the local authority, because this is very, very, very important, especially if you want to engage the small and medium scale industries, right? Okay, just to acknowledge our funding agency, of course, Ministry of Higher Education Malaysia for the Research University Grants, and MOSTI, Ministry of Science, Technology and Innovation Malaysia. RMC stands for Research Management Center, UTM. They are responsible for handling all our research grants, also definitely for all my students and my colleagues. Okay, these are some of the books which is available related to our work with heavy metals, with use of agricultural waste, with utilization of what we have around us. Yeah. So if you have the time, you can go through these books. Yeah. And just to show some slides of the people in UTM. Yeah, these are both staff and students. What else? Okay, with that, I thank you. These are my small research group. With that, I thank you very much. Thank you for your attention.
Thank you very much, sir. The session was very informative. We have a couple of questions here from YouTube, so I'll ask them to you, sir. Is that fine?
Yes, please.
The first question is, how can you maintain the pH level throughout the reaction, as the products produced will vary the pH in the bioreactors?
Okay, we have the pH control at the influent section, meaning the effluent pH is something that you cannot control, but you can always control the pH that will go into the bioreactor, which is much, much more important, because you want to control the optimum pH where your microbes will function. That's my answer.
Okay, thank you, sir. The next question is, what sort of pH electrodes are being used?
Which one?
I think this is for the chromium reduction process.
Oh, we were using the ERP. We don't, the pH of the electrode, we don't control. We are basing it on the influent pH. However, we set it to measure the ORP, what's the rate, yeah? It's some MV, some plus millivolt. I cannot remember the figure, but we set it against the ORP of chromium 3, so that we can know that when you got that value of a certain MV is on the plus the positive side, that chrom 6 has been reduced to chrom 3. It's like that. Yeah, we don't actually have a specific pH for the. We don't set the pH to suit the electrode's pH. Yeah.
Thank you, sir. The last question is, can any other fruit or bio waste be used for reduction of CR6?
Okay. Actually, before we decided to use liquid pineapple waste, we also have this idea of using tapioca waste. Okay, because in Johor we also have a big plantation of tapioca. Okay, the sago processing will produce waste which is rich in cellulosic materials. Okay, however, we at the end, when you want to bring this to a larger scale, cost will be of factor. Liquid pineapple waste we got it for 1,000 liter is only for 50 ringgit. Okay, I think it's around 500 rupees. Okay, for 1,000 liter, one meter cube. Okay, which is much cheaper compared to if you want to get it from the sago processing facility. Or the thing with agricultural waste is that if you want to use it in a small volume, fine, you can take it for free. But if you are talking about to take daily 1,000 liter, they'll start to charge you. that's when you have to really consider the types of agricultural waste that you want to use.
Thank you, sir. With that, we conclude the Q&A sessions.
Yeah.
Thank you very much for an informative and an in-depth session on using pineapple waste for reducing CR6 biofertilizer production and other biotechnology applications of microbes.
Yeah, good noon to all. Myself, Kalavati, I will give you the thank you note.
Okay.
On behalf of the organizing committee, International E-Conference on Frontiers in Industrial Biotechnology, I'm delighted to thank the speaker of the session 2. I'm thankful to our distinguished speaker, Dr. Zainul Ekmer Zakaria, Associate Professor, School of Chemical and Energy Engineering, Faculty of Engineering, University of Technology, Malaysia, for accepting our invitation to give plenary lecture. I'm sure that this lecture has given us a strong insight on the laboratory and on-site applications of bacterial technology for the reduction of environmental pollutants. I once again thank you, sir, for spending your valuable time with us and gracing us with your presence. I'm also thankful to all the participants for your kind cooperation throughout the session. Thank you, sir. Thank you all.
Okay, thank you all. Thank you for the invitations. All right, thank you very much. Bye-bye.
Yeah,
Bye,
sir.
thank you very much.
Thank you very much, sir. We will now take a short break and return at 2:30 p.m. IST for the next lecture by Dr. Jose Teixeira, who will be talking on cleaning technology for the production of chemicals. I request you all to log in 5 to 10 minutes earlier. Thank you. Hope to see you all then.